Okay, can you all see that?
Yes.
Awesome. Okay. Hi, everyone. My name is Sarah Anderson. I am a chemistry PhD student at Northwestern. I'm actually graduating in April, so I'm close to being done and very excited about that. Thank you. So today I'm going to talk to you about some of my research that I've done on accelerating the development of treatments for brain cancer. But first, a little bit about me. So I was born and raised in Denver, Colorado. I then went to my undergraduate at Santa Clara University, which is in the Bay Area in California. And then I came to Chicago to attend grad school at Northwestern. So yeah, I love chemistry. I also love writing and try to write about my science for non-scientists as much as possible. I have a cat named Mulligan, and these are my parents' dogs, but I consider them mine too. Their names are Rory, he's a golden retriever, and Wendell is a corgi. I love Disney, and I've been to every Disney park except for Hong Kong. That's the last one on my list. And I love Taylor Swift. This picture was actually taken at her Chicago concert that I was at, and it was a rain show, which sounds like it was going to be disappointing, but as you can see, Taylor handled it like a pro, and it was really fun. Okay, so now onto the science. You might have heard the word glioblastoma in news reports upon the decline and death of American politician John McCain. Mr. McCain was diagnosed with the aggressive class of brain cancer in July of, 2017, and he stopped treatment on August 24th, 2018, and he passed away the very next day. While this timeline might seem sort of exceptional, the unfortunate reality is that Mr. McCain's experience with glioblastoma is very accurate of the rapid progression of the disease. And in fact, those with glioblastoma survive on average just over 1 year after being diagnosed. So current treatments for glioblastoma include surgery and then chemotherapy. So a common first step with any type of cancer is to try to remove the tumor surgically. But the fact that glioblastoma tumors are in the brain makes it really complicated because if you don't remove the entirety of the tumor, the cancer will still be there and will come back. If you remove too much, however, you risk damaging healthy brain tissue and impairing cognitive function. So for this reason, the vast majority of glioblastoma patients who undergo surgery experience a recurrence of the cancer. And then the second treatment option is chemotherapy. Glioblastoma patients typically receive chemotherapy in the form of a drug called temozolomide, which causes the cancer cells' DNA strands to break and causes the cells to die. But with time, glioblastoma cells manage to adapt to become resistant to temozolomide, and it makes treating glioblastoma with chemotherapy really difficult. So with the goal of developing new treatments for glioblastoma, scientists examine glioblastoma cells for any irregularities that could be targeted by drugs. And they discovered that glioblastoma cells feature higher amounts of this enzyme called isocitrate dehydrogenase 1, or IDH1, relative to normal cells. So enzymes facilitate specific chemical conversions within the cell, and IDH1 helps to convert a molecule called isocitrate into a different molecule called alpha ketoglutarate. And at the same time, it helps to convert a separate molecule called NADP into another molecule called NADPH. And our cells use NADPH to produce precursors that help to build up the cell membrane. And so scientists believe that by having more IDH1, glioblastoma cells overproduce NADPH and are therefore able to divide more rapidly than healthy cells and ultimately form cancerous tumors. So now that we better understand the role of excess IDH1 in contributing to the progression of glioblastoma, is there anything that we can do about it? So the best way to counteract the effects of IDH1 in glioblastoma cells is to use an inhibitor, which is a small molecule that binds to the enzyme and prevents it from converting isocitrate to alpha ketoglutarate and NADP to NADPH. So identifying a molecule that can act as an inhibitor is kind of like trying to win the lottery. So if you were trying to win the lottery, ideally you would buy as many tickets as possible to try to increase the likelihood of winning. And similarly, scientists test millions of molecules to increase their chance of finding an effective inhibitor for IDH1. So in order to evaluate if a molecule is a good inhibitor of IDH1, scientists measure how much alpha-ketoglutarate is produced in the presence of the inhibitor. So the less alpha-ketoglutarate produced, the better the inhibitor. In order to measure how much alpha-ketoglutarate there is in the cell, researchers use an instrument that separates out all the different molecules that are in the cell to isolate and measure alpha-ketoglutarate. This is a very slow process. It's like if you had to count only the pennies in a pile of coins, maybe from your lottery win. And so you separated them into different piles of each type of coin and then counted the pennies. So evaluating all of the possible IDH1 inhibitors using this instrument is really time consuming, and it's delaying the discovery of a viable treatment for glioblastoma. So my research aims to minimize this delay by developing a tool that can measure alpha-ketoglutarate in the cell to evaluate IDH1 inhibitors, but that is also capable of rapidly analyzing a large library of potential IDH1 inhibitors. So the tool uses a plate that is coated uniformly with molecules, forming what is known as a monolayer. The molecules in the monolayer are designed to undergo a chemical reaction with alpha-ketoglutarate that captures it to the surface. The plate is then analyzed by mass spectrometry, which is an experimental technique that reads the mass or the weight of each molecule in the monolayer and produces a peak at its mass value in the resulting mass spectrum. The intensity or height of the peak corresponding to the mass of the monolayer alpha-ketoglutarate reaction product is proportional to the amount of alpha-ketoglutarate captured to the surface. So this method is faster than the technique traditionally used to measure alpha-ketoglutarate for two reasons. The first is that the chemical reaction occurs almost exclusively between the monolayer and alpha-ketoglutarate. Returning to our coin analogy, it acts like a special magnet that attracts only the pennies from the pile of coins so that you can isolate them and count them more easily. The second reason is that this tool can accommodate hundreds of samples at once. The surface of the plate is arranged in a 384 spot format that is compatible with robots that automatically dispense samples onto the plate. This means that up to 384 samples can be prepared and transferred to the plate at the same time. Analyzing the plate by mass spectrometry, which takes about 3 seconds per spot, provides mass information that can be used to calculate how much alpha-ketoglutarate was present in the sample applied to each spot. We use this method to measure the amount of alpha-ketoglutarate in 384 samples. And alpha-ketoglutarate could be detected in 377 of them, or 98% of them. It was very easy to do, and it only took about 30 minutes to collect the data, which shows that this tool is really useful for the rapid screening of large numbers of potential IDH1 inhibitors. So to assess the ability of the tool to accurately measure alpha-ketoglutarate in the cell, I used it to test cells that had been engineered to produce less alpha-ketoglutarate compared to 3 different types of normal cells. My technique, which is shown by the dark gray bars in the graph, measured a significant decrease in the alpha-ketoglutarate in the engineered alpha-ketoglutarate deficient cells. And I also used a more established experimental method to analyze the same samples, and those results are shown in the light gray bars on the graph. As you can see, the data line up really closely, which validates that our method is giving accurate measurements. So this is really promising and suggests that this technique could one day be used to assess the potency of IDH1 inhibitors for glioblastoma therapy. Because of the dependence on slow analytical techniques, long drug development periods are nothing new. In fact, the average time from drug conception to commercialization is about 12 years. But within that 12-year period lie almost 10 full cycles of glioblastoma diagnosis to death. So if there were ever a disease that could benefit from accelerated drug discovery, it's glioblastoma. So by developing a new tool that selectively captures alpha-ketoglutarate from many samples at the same time and quantifies alpha-ketoglutarate by its mass, I hope to reduce the amount of time needed to identify an effective IDH1 inhibitor and a potential treatment for glioblastoma. And that's all I have. I'm sorry that was quick, so please let me know if you have any questions.
I have a question to start us off.
Yeah.
Why is it called glioblastoma? Like, is it like each of those words means something? Because I feel like I've heard blastoma before, right? Does that mean
That's a good question. I mean, oma is
cancer?
often like a suffix for types of cancers. I'm not totally sure why, like carcinoma. And then glio, I'm assuming comes from like glial cells, which are in the brain. The
Okay.
last part, I'm not really sure.
I think blastoma has something to do with cancer, but I don't know for sure either.
No, that could be. I'm not totally sure.
Yeah, so I'm reading now. Glial is definitely a type of cell in the nervous in the brain, glial cells. Blastoma is a type of cancer caused by malignancies in precursor cells, and they're known as blasts. So I'm assuming the blastoma comes from the blast cells.
Very cool.
Thank you.
Yeah.
Sarah, looks like we have a couple questions in the chat.
Thank you. Let me, okay, so one person asked, what are your plans after you finish school? Great question. I'm actually planning to go to journalism school at Northwestern to become a science journalist. So not research. I'm going to step away from the lab because it's very difficult, although very important work. But I've just found that writing about science for different audiences is sort of more my passion. So yeah. Are you also concurrently working on treatments? So I'm not really working on actually developing those inhibitors. There are some labs at Northwestern that are doing that. There's a professor at the Feinberg School of Medicine called Alexander Stieg, and he's working on finding an inhibitor for IDH1. And they have a really promising candidate that we are currently discussing using this platform to assess it with, because yeah, they have found the other tools to be very slow. So there is potentially a promising candidate, and yeah, I'm hopeful that that will eventually be able to be developed into a drug. Are there other types of brain cancer? Yes, definitely. This is the most aggressive by far. And there's actually even different types of glioblastoma. So this is like what's called primary glioblastoma, which is when it starts in the brain, but it could start somewhere else in your body and then spread to the brain. And that's called secondary glioblastoma. And that actually, this everything I talked about with IDH1, that doesn't happen in that subtype. So it's really specific to not only like the type of cancer, but then like the subtype of cancer. And yeah, this is by far like the worst one, which is why we really wanted to focus on using faster screening tools for it. Yeah.
Are the other drugs or treatments or whatever it is that are used to treat glioblastoma now also targeting this same molecule?
Great question. No, they're not. They're just kind of going after DNA, just anything to kill the cells. They're not specifically targeting IDH1. For some reason, it's been really difficult to find an inhibitor that inhibits it. Sometimes that just happens. Another group at Northwestern, the Merkin group, is looking at spherical nucleic acids. So they have these compounds that basically introduce a certain type of genetic material that could stop the cell from producing IDH1. So instead of making it unable to be active, there would just be less of it overall. So that's another possible avenue. But the role of IDH1 in glioblastoma honestly was not even discovered until 2017. So it's still a pretty recent area that people are trying to target. Yeah.
Sorry, may I jump in if you don't mind?
Please.
Also, another problem with like, you also have to like think about the spatial conformity of tissues, you know, like for a glioblastoma, it's in the brain and, you know, you have to open up the brain. And also, I think a lot of drugs probably won't get in because we have something called the brain-blood barrier, which is very stringent and doesn't allow many things to go through. So even if you take oral drugs, like a lot of it probably won't go through your brain-blood barrier, and so it won't go through your brain.
Yeah.
So it's like, yeah, it's very difficult, especially when we talk about the brain.
Yeah.
It's very inaccessible.
I appreciate all your biology perspective.
Yeah, no problem.
Looks like we have one more question in the chat. Is radiation used as a treatment for
Oh,
this?
okay. Sorry, I just saw this. I think it can be. I know it's often used at least as a co-therapy in combination with surgery or chemotherapy. But it's, yeah, independently, I haven't read that it's as commonly used. And I don't know if that's, again, having to do something with the brain. There might be like some potential side effects of that. So I'm not totally sure, to be honest.
Yeah, sorry, another biology perspective. Yeah, radiation is commonly used as a co-treatment with cancer. You know, you usually excise the tumor and like you might not get all the cells, but radiation is a very aggressive and like not specific whatsoever. So radiation is targeted to kill fast-growing cells, which include, you know, cancer cells, but also, as I mentioned, like stem cells, which grow and divide very quickly. And that's why people lose their hair, because you lose the stem cells in your hair. And so radiation is very non-specific. It's just drugging you up with a chemical and hoping to kill the fast-growing cells. So I believe most oncologists do pair surgery with radiation. But yeah, it's very difficult. I think Northwestern actually has probably a new technology called proton therapy, which is like shooting proton laser into your brain, and that's a little bit more targeted. But still, like you can still get the other neurons in your brain. I'm not very informed on the cancer side of things, but that's kind of my general understanding of it.
Thanks.
Awesome. Any other questions for either Sarah or Catherine? Great. Well, thank you so much, Sarah and Catherine. You both did an excellent job, and we really thank you for being here and doing this with us. And thank you to everyone who attended. We hope that you'll join us again in the future. As I mentioned at the beginning, this is a monthly program, and we try to send it out. Our events as of now are the second Thursday of the month at this time, 1 o'clock, but we always try to send it out in the newsletters. So you should be able to find the Zoom links and the topics all in advance. So hopefully we'll see you again soon. Thank you, everyone.
Thank you for coming.
Yes, thank you. The questions were great.